Good afternoon, everybody. So still awake. The second last talk for today. So you're brave that you still make it. So today I want to introduce you to some of the data we achieved with regard to Periphylla, so the helmet jellyfish in a Norwegian fjord. This was done in the so first of all, the first introduction on Periphylla. So for those of you who are not familiar with this specific species, so this is a Scyphozoan jellyfish species, which has actually a holoplanktonic life cycle, which is then a direct development from the egg to the young medusae. And it's a species actually where the planula stage and the polyp stage and the ephyre stage are missing. So for Periphylla, this has caused quite some concern in Norwegian fjords because of quite abundant aggregations in many of the fjords since the 1990s. And this was considered to cause regime shifts from a fish-dominated fjord ecosystem to a jellyfish-dominated system. So here we wanted to gain further insights on the population dynamics of this more mesopelagic species usually, on its population structure and also the connectivity of Periphylla in Trondheimsfjorden. So for the methodological approach, what we did is, first of all, this whole data set is really based on a long-term data series which was conducted far more before my time when I started at NTNU in Norway. So this was a trawling campaign which were conducted in the framework of several projects actually. So it was the different funds which realized these trawl campaigns. And that was between 2006 and 2015. And thereafter, we did a 3 years trawling campaign during different seasons and years within the framework of the GoJelly project. So what we gained from these trawl campaigns was the biomass estimates for Periphylla in different areas of the fjord. And also during the last 3 years, so within the framework of GoJelly, we also got size distribution patterns. So just to introduce you to Trondheimsfjorden in general, so this is a bit more north than here. So it's still below the Arctic Circle, but it's midway up in Norway. So exactly here, there is Trondheim. And fjord ecosystems in general have the feature that they can be rather deep. So Trondheimsfjorden deepest station is around 600 meter deep. And what is characteristic for fjord ecosystems is that they have at least one sill, but many of them have often also several sills, which are considered as barriers in these ecosystems. So for Trondheimsfjorden, we have actually 3 sills. So there is one sill at the entrance, one in the mid-fjord, and one in the inner part of the fjord. And so also the fjord ecosystem is separated into 3 distinct basins. And we sampled Periphylla in all 3 basins in order to evaluate its population dynamics and connectivity. For the methodology, in addition to the trawl data, we also extracted DNA from 190 individuals of Periphylla, which is circa 15 to 30 specimens per location per sampling day. Small pieces of tissues were collected and preserved in ethanol, and then we used the CO1 as a marker. Overall, we must admit that the success rate was rather low, so only about like 50% success rate with a CO1 sequencing. But we ended up in having 93 Periphylla, good sequences of Periphylla in order to do this analysis. Thereafter, median joining networks were applied, and those then in order to figure out about the different haplotypes which we found within Trondheimsfjorden, but also adjacent waters. When it comes to the results, so first of all, here on the left-hand side, you see the catch data of peripheral biomass. And I must, the graphs which you see on this slide are now focusing only on the innermost part of the fjord, because the innermost part of the fjord was the one where we found the highest abundances. This doesn't mean that we didn't find periphylla in all other areas of the fjord, but the abundance were by far higher in the innermost part compared to the outer and middle fjord system. Up here you see the so-called catch per unit effort, which we calculated from the trawled catches. And you can see, so the data series started in 2006. And then there is a small gap here. And then we took up with the Go Jelly sampling in these 3 years thereafter. And in general, for the inner part of the fjord, you can see that there is quite some variation in biomass, which we caught during the different trawl activities. But overall, there was not really a general trend which we could find over the past 15 years of data. In addition, as I mentioned, we also did size distribution data, but only this is restricted to the last, those 3 years from 2018 to 21, where we also have size distribution data. And again, this is now only data for the innermost part of the fjord from 2 years, where we trawled in this area of the fjord. And you can see that between the 2 years, there were quite some distinct differences between the size distribution of periphylla, which we found. But in combination of the 2 years, we found all three or all major size classes of periphylla in this innermost fjord ecosystem, and especially also the young of the year, so the very early life stages of periphylla, which we found in quite some high abundances in 2020. And so this occurrence actually of the young of the year periphylla points then to a local reproduction in the innermost part of the fjord. In addition to this, we also performed population genetics. So what we found in general were 30 haplotypes, which we compared, so between the Trondheimsfjorden area and also some other Norwegian fjords. So the bluish color here is are the samples which were taken within Trondheimsfjorden, and the more greenish, brownish ones are from other areas of Norwegian fjord ecosystems. And what you can see is that we didn't really find a distinct population structure within Trondheimsfjorden over larger spatial scales, so along the Norwegian coast compared to the Trondheimsfjorden ecosystem. And so this means that there is a restricted gene flow between there is not a restricted gene flow between the populations of Trondheimsfjorden with adjacent waters. And this is mainly related so that the sills seem not to play big roles as a physical barrier for the population distribution of this species. So overall, the population outbursts of periphylla in Trondheimsfjorden were not confirmed. So this is, at least based on this data set, we cannot really see a clear trend. And also the poor geographic population structure actually provides evidence for a strong dispersal between the Trondheimsfjorden populations and the populations outside Trondheimsfjorden, which is maybe triggered by a frequent deep water renewal of this ecosystem, which allows a frequent gene flow. At the end, I want to mainly thank Jarle Mork, who established actually this long-term data series on periphylla, and his trawling activities and dedication of periphylla in Trondheimsfjorden. And I thank you for your attention. Thank you.